Hi everyone, so today we're going to be looking at A2.2, which is cell structure in IB biology. Let's get started. First, let's talk about cells. They are the basic building blocks of life. So all organisms are made of cells. Some are just one cell, whilst others are multicellular. So the fish, the moss, Listras, the lettuce that outlasted Listras in office, they're all made of cells. A 70-kilogram human has about 40 trillion cells, and each one will be specialized for different functions. And that's what the cell theory states, that all living organisms are made of cells. There's a few exceptions, but it's still considered to be true for the most part. Moving on, so microscopy. You need to know how to use a microscope. It's very hard for me to teach you that now. I hope your teachers have done that. If not, I'm sure there's some good videos on how to use a microscope, but what's very important is that you know how to use this formula, so how to calculate magnification, or the size of image, or the size of specimen. There are 4 steps. First, you need to convert all the measurements to the same units. This is if 2 measurements are given. Then you need to rearrange the equation if this is required. So if you're calculating magnification, you don't need to do this, but if you're asked for the actual size of specimen, then you have to flip them around. Then the next step is to measure the image size if required. So if this is not given and you need it to complete the formula, then you can just measure it with a ruler. And then finally, you solve the equation. Let's look at how this actually works in real questions. So this is a typical question. The size of a structure on an image is 3 millimeters and the actual size is 3 nanometers. Find the magnification. Okay, in this case, step number 1 is converting everything to the same units. So 3 nanometers is 3 times 10 to the power of minus 6 millimeters. You can use a unit converter online if you struggle with this, and you will learn it slowly, so don't worry. Then step 2, we don't need to do. Why? Because we're finding the magnification. That's already the subject of the equation. Number 3, we don't have an image, so we can't measure image size. So moving on to step 4, we just substitute in, and that gives us a magnification of 1 million times. Let's look at a harder one. The magnification on this image is 1,000 times. Find the actual size of the cell. Okay, here we don't need to do step 1 because there's no units given. We do need to do step 2, so we need to flip the equation so that we're actually finding the actual size of specimen, which is what we're interested in. Once we do that, step 3 is to measure the image. In this case, don't come for me, it's probably not 3.5 centimeters, I just made that up. And then step 4 is to just substitute in and solve, giving us 35 micrometers. For these exercises, it's very important to be well accustomed to changing units and doing these simple calculations. So if that's something you need some help on, I'm sure you can find some videos. And if you have any questions, leave them in the comments and I will respond. Now we need to talk about microscopy as in the theory behind it, right? The developments. So microscopes were first invented in the 17th century, so there's been a lot of change since then. Light microscopy has a lower resolution. I mean, it's still 0.00002 millimeters, so quite small, but you have to bear in mind that a lot of biological structures are very, very small. And then within light microscopy, this is your average microscope that you'll find in the lab, you can use fluorescent stains or immunofluorescence. So fluorescent stains are used to color specific chemicals in cells, such as DNA. But not all components can be traced with chemicals just because we don't have the chemicals to do it. So instead, you can also do immunofluorescence. This is where you use an antibody. If you don't know what an antibody is, don't worry, you'll learn that in the future. But an antibody just binds to something specific. So you can make it specific to the chemical you're interested in, and then the antibody itself will have a fluorescent marker. In that way, you are making what you're interested in fluorescent just indirectly, right, through an antibody. And then we have electron microscopy. So this is newer and it has a much higher resolution, actually 200 times better. So you can observe smaller structures. Within this, we have cryo-EM. This is quite complex, but it's basically used to determine protein structure. So studying the structures of proteins is much, much harder than you would think. And this is one technique in which you do it. You freeze the protein solution, and then with a microscope, you tilt the solution and use algorithms and computers. I also don't know how it works. It's very, very complex, but this creates a 3D image of the protein that then you can use. And then freeze fracture EM is for surfaces of cells. And what you do is you take a sample of cells, you freeze them, and then you break them, right? And then the rupture point, you can observe that surface by making a mold around the surface and looking at that through the electron microscope. Hope this is clear. You don't need to know extreme details, just know that light microscopy is older and less resolution. Electron microscopy can help us observe smaller components. Now let's look at some things that all cells have. The first is the plasma membrane. This encloses the contents of the cell. It controls the exit and entry of chemicals. It allows for a different environment within the cell than in the surrounding environment. And when the plasma membrane breaks, this is called lysis of the cell, and it always leads to cell death. The second is the cytoplasm. So the cytoplasm is mostly made of water. This links back to the first topic in IB biology, right? The soluble properties of water. It's because most reactions need to happen in water, and this is why the cytoplasm will mostly be made out of water. And the third is DNA. And this, as we've talked about in nucleic acids, just contains the information for the cell to carry out its functions, the genetic material. Now let's play a little game. So cells can be divided into prokaryotes and eukaryotes, but sometimes it's a bit hard to tell what's a prokaryote and what's a eukaryote, especially when just starting this course. So let's guess. I will let you pause for a sec and then try and guess what this is. It's a prokaryote, right? So bacteria, unicellular organisms, prokaryotes. What about a rose? A eukaryote. What about humans? Eukaryotes. What about fungi? Eukaryotes. So you can see a trend. Multicellular organisms are eukaryotes. Unicellular organisms, so one cell, are mostly prokaryotes. And this is an important distinction. Prokaryote cells, as we'll see, are a bit simpler. So these are the domains of life. All organisms can be classified in these three domains. And both bacteria and archaea are unicellular. And then eukarya, they're normally multicellular. So let's look at prokaryotes first. First of all, they don't have a nucleus and they don't have compartmentalization. What this means, the second word, is that there's no individual components with a membrane within them. So they're very small. Again, they still have a plasma membrane because all cells do. They also have a cell wall, actually, that you can see over here. Let me see if I can get the pointer, right? So they have a cell wall, and this is much thicker than the plasma membrane, and it protects the cell and maintains its shape. And it's made of peptidoglycan. And then you will observe that there's no compartments, right? No compartmentalization, so no organelles. There's only ribosomes, and these are 70S ribosomes, so they're different to the eukaryotes. We'll look at that in 1 second. Then there's also DNA, but it's not associated to proteins, and it's not in a nucleus. It's just free. It's called naked DNA. Okay, hope that wasn't too quick. You also need to be able to identify these cells and draw them. So again, ribosomes, the cell wall, the plasma membrane, the cytoplasm, and the nucleoid, which contains the naked DNA. Moving on. Eukaryotes also have a plasma membrane. Some of them have a cell wall, but not all of them. And they are compartmentalized. So they have organelles within them that have their own membranes. A lot of them. We will look at them in the next slide. There's also a nucleus, and this is where the DNA is stored in chromosomes. And the DNA is wrapped around histones. You can go to the nucleic acids video to learn a little more about that. But the nucleus will have a double membrane with pores in it so that things can come and go. And the DNA in this case is linear, whereas in prokaryotes, it's circular. We also have ribosomes, but in this case, they're 80S ribosomes. That's an important difference again. And other organelles such as mitochondria. These are most of the organelles that are found in eukaryotes. There's more, but these are the main ones. Starting with chloroplasts, these are only found in plants, so that's an important difference. Then we have the smooth ER, used to synthesize lipids, free ribosomes, which synthesize proteins, vacuoles, which are just filled with fluid, and these are bigger in plants. Mitochondria, the powerhouse of the cell, it helps with respiration, all right. The nucleus has the DNA. Golgi and the rough ER work together, so these two are better understood together. The rough ER is near the nucleus, and what it does is it has ribosomes on it, but instead of free ribosomes, they're attached to the ER, and they will produce proteins that then go to the Golgi, and then the Golgi can make them secrete, so leave the cell. So these are proteins that you'll need elsewhere in the body. I know that was really quick. Again, I can explain further in the comments if anyone's interested, but that's just a brief overview. So now onto the differences between different eukaryotic cells. So a cell wall is, plants and fungi have cell walls, animals don't. In plants it's made of cellulose, in fungi it's made of chitin. Plastids, only plants have them. You have chloroplasts, as we described, for photosynthesis, and amyloplasts that store starch. Vacuoles, all three have them. However, in animals, they're normally small and temporary, whereas in plants and fungi, there's normally less, but they're a lot larger. Then centrioles, these are organelles that are like a cylinder, that's why they're called centriole, and they organize the assembly and the movement of structures within a cell. So all animal cells will have them. Fungi and plants only have them in swimming male gametes, so reproductive cells. Very rarely. I put a tick, but in reality, most cells won't for plants and fungi. Same goes for cilia and flagella. Only male gametes have them in fungi and plants. In animals, they're a lot more common. Talking about the cell theory before, remember there were some exceptions. Well, here are some of the exceptions. There are some cells that don't have a nucleus. These are red blood cells. That's because these carry oxygen, so if they had a nucleus, they wouldn't be able to carry as much oxygen and they wouldn't be as flexible. And then phloem sieve tube elements, these allow sap to go through in a plant, so through the stem. So the lack of nucleus allows for the sap to flow through. And then there's also cells that have more than one nucleus, which again is an exception. So skeletal muscle, this is because these cells are formed by other cells fusing together, so the nuclei bundle, and then a septate fungal hyphae. Now let's talk about the processes that all organisms have to go through. In the case of unicellular organisms, since there's only one cell, that cell has to do all of these processes. There's a lot to remember, so I used ChatGPT to make this sentence so that you can remember it. So it's happy mice need every green rock regularly. Homeostasis, which is maintaining a constant internal environment in an organism. Metabolism, which is all the chemical reactions that occur in a living organism. Nutrition, which is just having the nutrients required for energy and growth. Excretion, which is removing the waste products of metabolism. Growth, which is an increase in size, or if you're multicellular, having more cells. Then the response to stimuli, which is perceiving stimuli and carrying out appropriate actions in response. And reproduction, which is producing offspring. And this can be sexually or asexually. But all organisms must do this. Let's do a question. This is style paper 1 question, and it's which of the following are structures found in prokaryotes? Again, pause now. I will give the answer in 1 second. So the answer is C. 7ts ribosomes, nucleoid cell wall. Remember, 80s ribosomes are in eukaryotes, 7ts in prokaryotes. So B and D are not the answer. And then remember, prokaryotes have no mitochondria because they don't have organelles. They're not compartmentalized. Let's talk about the higher level content. This is a bit more complex, of course, but starting off with endosymbiosis, this is the idea that, so endo stands for inside, right? So one organism living inside another. And symbiosis is when two organisms collaborate. So this is how it is thought that eukaryote cells came to be from prokaryotes. Prokaryotes are simpler, so they were there before, it is assumed. But how did eukaryotes came to be? Well, it's thought that mitochondria were prokaryotes themselves, which could have aerobic respiration, right? So they could produce energy through aerobic respiration. They would have been eaten by another prokaryote, but then instead of being digested, they just started living there. Why? Because it was beneficial for both. The prokaryote which did the aerobic respiration provided energy for the host, and the host could provide food to the one that did aerobic respiration. So again, it was beneficial for both, so it just stuck like that. And then chloroplasts would have been similar. A prokaryote which was able to do photosynthesis would have been engulfed by a bigger one, and it would have just lived there and survived. What's the evidence for that? Well, both mitochondria and chloroplasts have 7ts ribosomes inside them, like prokaryotes. They have naked circular DNA, like prokaryotes. They can replicate by themselves without the cell, like prokaryotes, and they have double membranes, like prokaryotes. So it does make sense that this would have been the case. This is not proven. It's impossible to prove because it happened millions of years ago, but this is what is thought to have happened, and this is the theory of endosymbiosis. Perfect. Let's do another question. What are the following structures? Again, pause now. The answer is that one is a mitochondria. You can see the cristae and the invaginations, so the double membrane as well, that's common to mitochondria. And then the second is the Golgi apparatus. You might have thought it's the rough ER. The way to distinguish that is that the rough ER is by the nucleus normally, and this is not. So again, the ER processes the protein, sends them to the Golgi, and then the Golgi is further away from the nucleus, and then it sends them away from the cell. Perfect. And then the last two things. So it's important to know that in multicellular organisms such as us, different cells perform different functions because this is more efficient, right? Instead of having all cells do everything, you can have them be specialized. How does this happen? Through gene expression. So what this means is that not all the DNA is turned on all the time, but rather you turn on some genes in some cells and some other genes in other cells, and that leads to specialized tissues, such as immune cells, sex cells, muscle cells, fat cells, etc., etc. And so it's thought that multicellularity has evolved many times from unicellular organisms. So what are the advantages? Well, first, a larger body size, obviously, right? If you have more cells, you're going to be larger than if you have just one. Then specialization and complexity, because as we just talked about, you can have cells to do different things. And then also, if one cell dies, the whole organism doesn't die because you can just generate more cells. So you have a longer lifespan than a unicellular organism. However, it's important to note that there must be some advantages to being unicellular, given that most organisms on planet Earth are still unicellular. Okay, with that, we'll finish here. This is the end of the topic. I hope everything was clear. Again, leave any comments if you have any questions, and I'll see you next week for the next topic.